All right. Hello, everyone. Thank you all for continuing to join us throughout the day here at the Lytham Partners Fall 2024 Investor Conference. My name is Robert Blum, Managing Partner at Lytham Partners. And during this presentation, we welcome Shuttle Pharmaceuticals, ticker symbol of SHPH on the NASDAQ. And joining us today from the company is the Chairman and CEO, Dr. Anatoly Druchilo. Before I turn it over to Dr. Chillo, I want to remind everyone that management is available for one-on-one meetings throughout the conference here today. If you've not already signed up and would like to schedule a one-on-one, you can send me an email. That's Blum, B-L-U-M, at lythampartners.com, or you can visit the website for the conference. That's lythampartners.com/fall 2024. From there, you can click on the investor registration button to make your one-on-one selections. With that, Dr. Chillo, thank you for joining us, and the floor is all yours.
Good afternoon, Robert, and thank you for the introduction. I'd like to share with the audience some of the events that have taken place with Shuttle Pharmaceuticals Holdings in terms of our reaching certain milestones in our efforts to develop drugs for radiation therapy sensitization. This is our forward-looking statements. You can look at this on the deck later. The fact that we are trying to, the main goal of our research and the focus is to improve the results, the outcomes of patients that undergo radiation therapy for cancer. The fundamental problem is that the dose that we can deliver these days is terrific. We can shape the radiation, we can deliver high doses wherever we want in the body, but our limitations still consist of the tolerance of the normal tissues that are either surrounding the tumor or are integral with the tumor. And here we see several types of machines. It's a conventional radiation therapy linear accelerator, and it's a proton machine. And as you can see, the CT scans that are shown show that very high areas of dose can be achieved, but then there are areas of normal tissue that need to be spared. So there are two ways that we can approach this. Shuttle Pharmaceuticals is developing sensitizations, a sensitizer to make the cancer cells more sensitive to the radiation effects. And then there's the activation of the immune system in order to stimulate the immune system to attack some of these cells after radiation has caused the release of certain antigens to the surfaces of the cancer cells. I will address our initial work, which is with sensitizers. And I would say to you that the concept of sensitization is established in our field, but most of the drugs that are available are currently used off-label. And the one that we're developing is a drug that has no other effects. It only works with radiation. The company overview shows our structure, which includes the parent company, which is a holding company, and we function with the diagnostics as Shuttle Diagnostics Incorporated, and we're placing those into those intellectual properties into that company. And Shuttle Pharmaceuticals is the therapeutics and development of the therapy molecules. The key molecules are rupedex-uridine, a clinical stage orally administered cancer radiation sensitizer, and the HDAC6 inhibitors. SP225 is the one we have selected to develop for stimulating the innate immune system and to activate the immune response after radiation. We have two diagnostics intellectual properties. Both of these were developed by our scientists in collaboration with other scientists at Georgetown University. And we have developed the PC-RAD test, which is a blood test for predicting outcomes of patients who undergo radiation therapy for localized prostate cancer. And then the PSMA ligand is a theranostic molecule that offers diagnosis and therapeutics for patients with prostate cancer. The ligand is a small molecule that binds to the surface of the PSMA antigen, which exists on prostate cancer cells. So how does the therapeutic agent ropedexirdine work? Well, ropedexirdine is a prodrug of the drug called IUDR, and this is the active ingredient. This is a powerful radiation sensitizer developed by the National Cancer Institute more than 30 years ago. And following oral administration, the prodrug, which is ropedexirdine, it's converted to IUDR, and it has the ability to get into the DNA and then make the DNA more sensitive to the radiation effects by increasing the number of free radicals to break the DNA. The drug itself, as you can see, has been tested, the active ingredient, uh, back with phase 1 and phase 2 trials with radiation therapy alone compared to radiation plus IUDR. And you can see a significant increase in the median survival for both astrocytomas and for glioblastoma. These were small studies. The drug had a problem with constant infusion delivery because it had a very short half-life. It had to be delivered by a pump over a period of 6 weeks. There were many issues associated with that, so that the development of the prodrug was a major improvement because now you can take a pill every time you get your radiation therapy. So the phase 1 clinical trial was funded by the NIH, and we performed it under a subcontract to Brown University Rhode Island Hospital, and that has been published in Clinical Cancer Research. And as we saw, we established the fact that the drug could be given safely to patients and had a maximum tolerated dose of 1,200 milligrams daily for 28 days. The next step is to go advance from the phase 1 to the phase 2, combining ropedexirdine with radiation therapy. We selected this IDH wild type methylation negative glioblastoma as our patient population for testing the drug. This is pretty much the most aggressive brain tumor that there is. About 50% of patients present with this type of tumor, and the standard of care is radiation therapy. The other drugs, such as temozolomide, really do not work in this particular type of tumor. We have manufactured the drug. We formulated at the University of Iowa Pharmaceuticals. We engaged Theradex Oncology, and for the last several months, we have been making sure that we had the right cancer centers. We selected 6 East Coast cancer centers to enroll patients. IRB, Institutional Review Board approvals have been obtained. The CRO services for the multi-institutional clinical trial have been engaged at each of these institutions. And we have 2 that are ready to start enrolling patients, the University of Miami and the University of Virginia. They completed all negotiations and are ready for patient enrollment. We anticipate enrollment of a total enrollment of 54 patients, valuable patients, and this is anticipated to take about 12 to 18 months, with an early readout and is anticipated in about 24 months. The second category of drugs that we have developed are the selective histone deacetylase inhibitors. These are epigenetic molecules, and we have a proprietary technology that we use to manufacture many of these using something called an ATM activation domain, which is a molecule that stimulates a gene called ataxia telangiectasia mutated, which is related to radiation protection. And then the zinc binding domain inhibits the enzymes that we're working with. And we found that we had several categories. We had the pan-HDAC inhibitors, which are useful as radiation sensitizers and normal tissue protectors. Then we had the class 1 HDAC inhibitors inhibiting selectively HDAC1 and HDAC3, and these inhibit breast cancer, especially estrogen receptor positive breast cancer growth. And then the class 2B, which is the SP2-225, which activates the innate immune system. The class 2B works with a mechanism of stimulation of the immune system through the macrophages that are indigenous to the tumor and to surrounding tissues. So at this point in time, we've selected SP225, the HX6 selective inhibitor, as our primary molecule for preclinical development. And then the 303 is for breast cancer, and we are looking for a collaboration to help develop that. The HX6 selective mechanism, it was shown here with this is some data from our collaborators at Georgetown University. We have an immune system that is stimulated by the fact that the tumor is irradiated and damage to cells occurs. Not all tumor cells are killed with the radiation, and the ones that are not killed then are attacked by the immune system through a process with the macrophages and giving you a selection against these cells. As we can see, we can inhibit growth of the tumors substantially in an animal model. This work has been published by our colleagues at the university, and you can see this data. The therapeutics, that we are developing are both clinical and preclinical. And we basically, as I mentioned, our main strategy focuses around the phase 2 clinical trial of ropedoxuridine and radiation therapy for brain tumors. Our preclinical development focuses around the HX6 selective inhibitor for immune stimulation. And then we have other drugs, including the class 1 HDAC selective inhibitor for developing, in each of these, we need to scale up the production of the drug and then to test it by collaborating with investigators in model systems that would be used in animal systems for the treatment of breast cancer. Now, the other part of the company, the diagnostics, and I would present it as a theranostics, but the initial part is diagnostics, is a test that we developed by looking at patients who had radiation therapy for prostate cancer. There are approximately 66,000 patients a year that undergo localized radiation for prostate cancer. And of these, the majority do benefit, but there are a group of patients that do not benefit from the radiation, but and would possibly be treated with other modalities, including surgery or perhaps with hormone therapy. So it's important to predict who would be able to respond well. So the diagnostic test, there is a substantial market for this, but there are no predictive assays that are available to determine success of a specific treatment. So this would be a first of its kind, and there's a key unmet need that we'd be addressing. The test has been developed with funding from the government through contracts for developing these assays. It consists of a number of vials with standards and comparisons in order for us to be able to determine the levels of certain metabolites that are present in the blood. And with this test, we would then have a readout saying whether the patient is at high risk for complications or low risk, or high risk for recurrence or low risk. Okay. The second area that we have pursued is the PSMA B that we developed. The PSMA is the prostate-specific membrane antigen. It is located on prostate cells and on prostate cancer cells. The development of a ligand was initially performed by a scientist at Georgetown University about 25 years ago. And that work has subsequently led to development of imaging agents and therapeutic agents that have been developed to the level of clinical applications through a large pharmaceuticals. The PSMA ligand that he designed for us was specific for the use in the treatment of patients with prostate cancer that would be treated with protons. And the idea was to develop this with boron and to do boron neutron or boron proton activations. So both of these are areas that are of interest to me personally as a radiation oncologist and to other radiation oncologists in order to improve the local treatment. But we also found that our molecule was very avid for binding to the PSMA, and by attaching radioactive sources to it, one can either develop an imaging agent or you can develop a therapeutic agent which is specific for metastatic disease and for prostate cancer. So this is the overview. So the receptors, when they bind to, they dimerize when the ligand binds to it. They dimerize, they're internalized, and the radioactive, source is then deposited to damage the DNA and to treat patients. The same process could be used to deliver other kinds of drugs also, but that would be for future directions. So here is an example of what's being done now. And the market for this global market is in excess of potential is in excess of $2 billion a year. The drug that was developed was this Pluvicto. And what happens is when smaller concentrations are injected into patients and then imaged on a PET scanner, and then one can get an image that shows you all these hot areas where the PSMA was present and is taken up into various spots in the bone and in lymph nodes, the ribs, and so forth. And so you actually have a way of delivering the therapeutic directly to the cancer cells, even though they're at a site that you normally can't target with anything but chemotherapy. And you can actually use it to deliver higher doses of a substance. So this has been developed using this lutetium PSMA construct, and the lutetium delivers the radiation to the tumors in these areas. And this has been shown to prolong life in patients with metastatic patients, or with metastatic prostate cancers. So we have these two areas: diagnostics, therapeutics, and then we can add to that the several approaches that I described. And I'd like to summarize for you briefly what the intellectual property underlying these are. We do have, for the ropidoxuridine, the drug itself has orphan disease designation. We have a method of use patent for the next generation that combines the IPDR with Tiparasil, which is a new formulation to increase the blood levels. And then, of course, we have manufacturing patents in process. The selective HDAC inhibitors, we have more than 20 composition of matter patents in the US, Canada, and Europe. These have been awarded to us. They were developed by scientists at Shuttle Pharmaceuticals, so we do not owe any milestone or royalty payments. And we've selected two of these, the 225 and the 303, as molecules for further development. patents also are available, for backup molecules and other selective molecules that we have not fully evaluated yet. The PC-RAD test, the predictive biomarker for prostate cancer, response to radiation therapy. This is a metabolomics-based diagnostic test. It was developed while I was at Georgetown University, and we have the exclusive license for the intellectual property. The PSMA-B, theragnostics and diagnostic capability. We have a unique molecule that has intellectual property associated with it, and the diagnosis and therapy. We've obtained an exclusive license from the other scientists that were involved in the development of this with the Shuttle Pharmaceutical scientists. The clinical strategy that we are looking for, the diagnostics would be essentially to test the predictive biomarker in a context of use for FDA approval. That would be basically to do a study of 360 patients who receive radiation and to follow them after they complete the radiation to monitor for cancer recurrence and for side effects, and then correlated with our test results that are predictive at the start of treatment. For the development of PSMA-B intellectual property, we are looking to collaborate with institutions that have strong nuclear medicine programs and to develop the diagnostic imaging. The therapeutics we will develop with our collaborators at Georgetown University for proton therapy testing. This is our overview of the financial position of the company. We currently have a small number of outstanding shares, and RSUs are restrictive stock units. So the total fully diluted basis is 2.35 million. The stock price at this time is, in my opinion, at a very bargain. At this time, we have a market cap that shows we'll need additional funding to finish all these studies. And this is our position as of today from our CFO. The leadership team, I list some of the credentials, but in terms of the clinical credentials, we have myself and Tyven Rich. We are both experienced radiation oncologists. We've served as chairman of departments. We've performed research and have several hundred publications to document our success. And I've served as both department chairman for many years, medical director of Georgetown University Hospital for 3 years, and interim director of Lombardi Cancer Center for 2 years. So much of the work, the administrative work, is similar in scope to what we're doing with the company in terms of developing this discovery and development of drugs. Dr. Mary Young is our biology expert, and she's a radiation biologist and an expert in performing laboratory and tissue culture studies. Peter is our CFO, president CFO. He manages the administrative aspects of the company. Michael Vanderhook is the vice president of regulatory. He was the former administrative director of Lombardi Comprehensive Cancer Center. And he manages the interactions with our CRO and our subcontracted cancer centers, many of which he actually knows many of the people involved. And then Tim Lorber recently joined us. He's a CPA with greater than 40 years of experience. He was managing chief accounting officer for Legg Mason for many years. And he's working with us to advance our company in terms of the financial status. We have a board of directors of, five people: Steve Richards, CPA and MBA, and he's a businessman who who uh, is the chairman of our audit committee. Milton Brown is a medicinal chemist and vice dean of research, East Virginia Medical School. I worked with Milton Brown for over 20 years. Betty Jacobs is a former dean of the School of Nursing at Georgetown University, and she's a distinguished scholar on and is involved in national and global health law. Chris Sanyaki is the CEO of TCG Green Chem. They manufacture drug, and we have a strong relationship with them. And Josh Schaefer has been a member of Big Pharma and helps us with our strategic planning. And that is about it. Thank you very much for your attention, and I'm glad to take any questions.
Look, we obviously, yes, if you have any questions here for Dr. DiCillo, please, reach out. We'll look to get a one-on-one scheduled for you. Again, you can send me an email. That's Blum, BLUM, at lythampartners.com. Again, you can also request a one-on-one meeting with Dr. DiCillo here throughout our conference. The landing page for that website, again, is lythampartners.com/fall 2024. From there, you can click on the investor registration button. Or again, if you'd like to schedule a meeting, just feel free to reach out to me via email, Blum again at lythampartners.com. So Dr. DiCillo, thank you very much for that informative presentation. Look forward to speaking with you again here in the future.
Thank you.